Our next speaker is Swapna Uplekar. Hope I pronounced it correctly. Swapna is a principal scientist in the genomics and sequencing program at FIND. And FIND is a global health nonprofit based in Geneva, Switzerland, with a mission to ensure reliable access to diagnosis across the world. So Swapna has trained as a molecular biologist and bioinformatician in the UK, with Switzerland and the USA. And she has worked with next-generation sequencing technologies for the last 15 years, working on the genomics and transcriptomics of tuberculosis to build genomics capacity for surveillance of malaria pathogens in limited resource settings. At FIND, she works to establish the use of next-generation sequencing as an in vitro diagnostic and surveillance tool in low and middle income countries for tuberculosis, AMR, COVID-19, and other outbreak-prone diseases. Thanks.
Thanks for that introduction, and thanks to Oxford Nanopore for the opportunity to present at this symposium. So I'll change gears a bit, and I'll talk more about the utility of targeted next-generation sequencing as a diagnostic tool for drug-resistant TB. And thanks to Justin for the background initially. And so I don't think I need to stress to this audience why TB remains a global health priority. It's still the world's top infectious killer. You see the staggering numbers on the slide from the latest global TB report. And as you know, Mycobacterium tuberculosis, the causative agent of TB, has acquired resistance to all the drugs introduced against it in the last several decades. And majority of the drug-resistant TB cases still remain undiagnosed, and even those who are diagnosed are only 2 out of 5 are estimated to receive treatment. The WHO End TB strategy calls for universal access to comprehensive drug susceptibility testing, or DST, for all persons with any form of TB. And this is to ensure early detection and to determine the appropriate and effective treatment for TB patients. Now, drug resistance in TB has evolved over the years, depending on resistance to one or more first or second-line drugs. You have monoresistant, multi-drug resistance, or extensively drug-resistant forms of TB. That said, there are more than 20 drugs currently used against TB in different combinations under different circumstances. In 2022, the WHO published landmark improvements in treatment options for drug-resistant TB, featuring some new and repurposed drugs. And in some cases, the duration of the regimens was brought down from 18 months to 6 months, which was great progress. So I've just listed some of the regimens of different types of TB here. And while the details are not relevant, I want to demonstrate the complexity of tackling DR-TB and emphasize the need to preserve the efficacy of these drug regimens through timely and comprehensive DST for patients. So conventionally, most of you will probably know, TB DST has relied on phenotypic methods, which is just based on the observation of growth or inhibition in presence of a drug. While these methods are comprehensive, they have rigorous lab requirements, especially for TB. And given the high slow growth rate of TB, these are highly time-consuming. So it can take, on average, 6 to 9 weeks to get any DST results. Now, as it happens for Mycobacterium tuberculosis, most of the resistance is caused through spontaneous mutations in the bacterial DNA. So this makes molecular methods and genotyping highly effective for detection of resistance. Now, most available rapid tests only detect a few key mutations in one or more genes, so they can only predict resistance to a few drugs. And if you want to detect resistance to all of the existing TB drugs, you would need to interrogate hundreds of mutations. So clearly, next-gen sequencing is ideal for comprehensive molecular DST of TB. Now, we would want to obviously go with whole genome sequencing, but even today, it works best only with MTB DNA extracted from culture isolates, so which does not make it suitable for timely detection of drug resistance. Targeted sequencing, on the other hand, has been shown to work directly from clinical samples and can allow detection of resistance to almost all drugs in a single assay, And depending on the technology, it can be done less than a day, in less than a day. So there's no doubt that tNGS-based DST could drastically improve clinical outcomes for drug-resistant TB patients. So why is it not being used yet? Now you must be aware that, the global burden of disease is disproportionately carried by low and middle income countries, and most of the emerging diagnostic and treatment options are available largely in high income countries. And genomics is affected by such global health inequity. So we started leading the Seq&Treat project, which was funded by Unitaid and was initiated in 2019 to address the key barriers that currently limit the utility of sequencing for TB patient care in LMICs. So this was led by FIND in collaboration with the WHO, and the project was divided into 4 main outputs. The first 2 outputs would generate the evidence and knowledge base required to support WHO policy on the use of tNGS. And outputs 3 and 4 would identify approaches to boost the adoption of commercial sequencing solutions for rapid DST. So today's talk will focus on the work that was carried out under outputs 1 and 2. Now, one of the reasons for limited uptake of sequencing in LMICs has been the perceived complexity of this technology. So the need to piece together disparate components and validate them individually. But if packaged appropriately, these can be transformed into a complete end-to-end solution, which includes optimal automation of intermediate steps and minimum requirements for highly specialized expertise to run the workflow from sample to result. When we started in 2019, there was no such existing pipeline of standardized and validated commercial tNGS products that could be utilized for DR-TB diagnosis. At the same time, being cost-effective and suitable for utility in low and middle-income countries. So we started the technology selection process based on the target product profile that was published in 2018 in the WHO Technical Guide. Solutions that met these criteria were selected for analytical validation. I'll describe it a bit more in the next slide. Based on the technical performance in this analytical validation, the solutions were then included in a multi-center clinical evaluation. And data generated from these studies was then submitted to the WHO for review to develop a policy recommendation. Now, what took me 30 seconds to describe has taken over 5 years to complete, and it's taken tremendous amount of work from numerous partners to go from a target product profile to a WHO recommendation on use of tNGS technologies for clinical diagnosis of DR-TB. So that's just one part of it. In addition to evaluating the diagnostic accuracy of targeted sequencing, through Seq&Treat, we also initiated the development of a clinical knowledge base hosted at the WHO to manage global sequencing data and associated phenotypic data for MTB isolates. The primary use of this data has been to develop a catalog of TB mutations to harmonize the interpretation of sequencing data, not just sequencing, but any genomic data from sequencing and other molecular tests. And we hope that it can act as an international standard that can be referred to by TB programs, researchers, clinicians, and industry partners. So the first version of this catalog used data from 38,000 isolates from across 40 countries. It was published in 2021 and listed over 17,000 mutations and the strength of their association with phenotypic resistance for 13 anti-TB drugs. Within 2 years, as more data became available from an additional 15,000 isolates, a second version of the catalog was published. This was in November of last year, with an emphasis on new and repurposed drugs, such as bedaquiline, clofazimine, and delaminid. So building on all of this Seq&Treat framework that was set up, we initiated a 2-phase evaluation of end-to-end tNGS solutions. One of the 2 solutions evaluated was the Ampor TB solution that Justin described earlier. The other one was based on the Illumina sequencing platform. And both solutions completed a phase 1 analytical validation, which was conducted using a blinded reference panel to validate the dynamic range and the SNP calling accuracy. And both solutions successfully completed phase 1 and then went ahead to phase 2, which was a multi-center clinical evaluation conducted in 3 countries, India, Georgia, and South Africa. And the objective here was to assess the diagnostic accuracy of tNGS at intended settings of use against the TB drugs listed here. And I've given some details of the study here, but just going through it briefly, the results were compared to a composite reference, which was a combination of phenotypic DST and whole genome sequencing. At the time the study was conducted, only version 1 of the Mutations Catalog was available, and that was used to interpret the data for drugs included in the catalog. And then, as per the usual WHO guidelines development group or GDG process, the WHO commissioned a series of systematic reviews to look at all of the published and unpublished data on these tNGS solutions related to their diagnostic accuracy and other evidence on economic and qualitative data on feasibility, acceptability, and so on. So Seq&Treat, the data generated in Seq&Treat formed a major part of the GDG review, as a clinical evaluation at this stage was never done before for TB. So before getting into the recommendation, I'll just give a brief summary of the results of the clinical evaluations. One of the key questions we wanted to answer with this was, can we sequence MTB directly from clinical sputum sediment samples? So here we have data for the AMPOR-TB assay, showing sequencing success stratified by the GeneXpert MTB-RIF result category as a proxy for a bacterial load. So on the y-axis, you have the bacillary load represented from high to very low, depending on the CT or the cycle threshold values of the GeneXpert MTB-RIF. And on the y-axis, you have the percentage of success, so to say. So AMPOR-TB showed a very high success, sequencing success rate. Over 80% of samples with high and medium bacterial loads produced full profiles with coverage across all the targets. Majority of the sample failures were in the very low and low bacillary load samples. On average, it was about 30%. But overall, there was 95% success of the clinical samples. 95% samples produced data with at least partial drug resistance profiles. So that was quite promising. Next, we can look at the diagnostic accuracy of AMPOR-TB. I'm showing the sensitivity data here, and there's a table on the next slide, but you can already see from here that AMPOR-TB solution showed very good sensitivity for most drugs, except the new and repurposed drugs. Aside from these 2 drugs at the bottom, all of the drugs listed above were compared to a composite reference of whole genome sequencing and phenotypic DST. For the new and repurposed drugs, as there were no mutations included in the version 1 of the catalog, the reference here used was only phenotypic DST. And it appeared that the reduced sensitivity may be mainly due to omissions of targets for these drugs in the mutations list that was utilized by Oxford Nanopore in this version. So overall, AMPOR-TB produced accurate and reproducible sequencing results that could be used for reliable detection of DR-TB directly from clinical sputum sediment samples, and supported the diagnosis of a wide variety of clinical drug resistance profiles. Now, when we look at the performance across the various drugs that are listed here, in green I've shown that the drugs for which the assay met the performance requirements, so these 7 drugs, 5 of these has a sensitivity of over 95%. The 2 drugs showed in gray are not recommended by the WHO anymore, so I won't talk about them. And then in red are the drugs that showed much lower performance. And for some of these, data could improve with an expanded mutations list, as the gene targets associated with resistance to these drugs are already covered by the assay. Now, that said, the AMPOR-TB assay is now included in the list of tNGS solutions recommended by the WHO for TB DST. So in March, so just last month, in the third edition of the guideline, the consolidated guidelines on TB diagnostics, the WHO for the first time includes targeted sequencing in the class of technologies that can be used directly on respiratory samples for people with bacteriologically confirmed TB as well as rifampicin-resistant TB. And the drugs for which the recommendation has been provided are listed in italics here. And I mean, one of the key points to highlight here was that this was the first time that tNGS was recommended rather than culture-based phenotypic DST. And it has been a breakthrough for the utility of genomics in infectious disease diagnostics. So, as the WHO GDG came prior to the publication of version 2 of the Mutations Catalog, as soon as it became available, we reanalyzed the reference data from Seq&Treat phase 2 using the new version of the catalog. And basically, we wanted to know how the WGS performed compared to the phenotypic DST using versions 1 and 2 of the catalog. And the biggest improvement came as expected for the new and repurposed drugs. As you can see, in green are the improvements for linezolid, clofazimine, and bedaquiline sensitivity. And we hope this would improve even further in the next version of the catalog, because even today, the data available for these drugs is pretty limited. The proportion of resistant samples in our data set at WHO was only 2% for clofazimine and 7% for bedaquiline, whereas for other drugs, we have around 25 to 30% samples that are resistant. So following the release of Mutations Catalog version 2, the ONT team also swiftly revised their bioinformatics pipeline, and we are currently reanalyzing AMPOR-TB data for phase 2 samples and processing it with the updated version of their pipeline. But initial findings already look promising and show substantial improvement in bedaquiline sensitivity from 6 to 69%, and for clofazimine from 0 to 67%. So the analysis is still ongoing, and I can't share more of the data, but it's evident from here, with this, and what I would like to highlight is that NGS is a highly flexible and adaptable technology, and as we strengthen our knowledge of TB mutations associated with drug resistance, we can show quite rapid improvements in performance of these solutions without any changes to the wet lab component of the assay, as long as the key drug targets are already included. So what I described so far puts in place the knowledge and evidence and policy needed to establish NGS use for TB clinical care. But policy can only achieve its intended outcomes when it's implemented effectively. So it's important to consider findings from the other assessments that contributed to the WHO recommendation. And I'll just go over these briefly. So broadly, targeted sequencing was found to be comprehensive, convenient, rapid, and adaptable. We are already, I think, quite well aware of the challenges associated with sequencing implementation in general, particularly the high cost and technical complexity. But despite that, tNGS was found to be cost-effective, depending on the context and the various other factors that are listed here. Other challenges included, the need for specialized infrastructure, expertise in wet lab and data management, issues related to procurement, especially in low and middle-income countries. But despite all of this, tNGS is still found to be acceptable and implementable in routine programmatic conditions in countries with high burden of drug-resistant TB. What is needed is, further investigation of optimal implementation models, ideas on placement of targeted sequencing in diagnostic algorithms, and other context-specific operational considerations. So as a companion to the diagnostic guidelines, just for your information, the WHO also updated its operational handbook, and now it includes some recommended algorithms for placement of tNGS-based DST for drug-resistant forms of TB. And last but not least, I think I'll end with just some key considerations for implementation that are listed in the operational handbook. So while So tNGS can be considered as an alternative for comprehensive DST due to its rapid turnaround time, especially when compared to phenotypic DST, but it's not intended to replace the rapid molecular tests. Initially, targeted sequencing is said to be more suitable for use at central or reference laboratory level and should be prioritized for samples with high bacillary load or in cases where the pre-test probability or risk of resistance is high. tNGS is recommended to guide clinical decision-making while understanding that more data is needed on the new drugs and the impact of this technology on patient-important outcomes. And while cost and feasibility for implementation in low and middle-income countries are potential barriers, there are opportunities to leverage the investments and NGS capacity that was built during the COVID-19 pandemic. So I'll end my talk here, and we are now in the next phase of the Seq&Treat project to support countries with adoption of tNGS and help them address critical gaps in drug-resistant TB diagnosis and DST, especially following the rollout of these new and repurposed treatment regimens. And I hope we have even more good news to share by this time next year. Thanks very much for your attention, and I would like to acknowledge the fine team, our donors and country partners that have made all of this work possible. Thank you.
Yeah.